Okay, now, so we see what ribosomes are like, how they're made. Let's take a look at one last little aspect of this before we actually look at the translation process itself. Now, most of you guys at least should remember this, okay? Anybody who's taking genetics, you should know this, but just in case. What is a codon? A codon is a sequence of 3 bases on an RNA. Say like U equals for instance, a sequence of 3 bases on RNA UUU represents the amino acid phenylalanine. That's one of the things that goes into NutraSweet. Okay? All right, so you have 3 bases, and the ribosome recognizes as, say, this means put a phenylalanine at this position. So in a sense, the language of RNA, of nucleotide sequence in RNA, is very much like a language, except in this case we only have 4 letters in our alphabet. That's A, C, U, and G. And all the words in our language are only 3 letters long. A lot more simple than English, right? Okay, but, so of course you have, since you can choose any 4 bases at any of these positions, it means you have 4 times 4 times 4, 64 possible codons. Okay, now, we got 64 possible codons. 3 of them are stop codons, it gives us 61 left. One's the initiation codon, which actually does code for an amino acid, by the way. It codes for the amino acid, the sulfur-containing amino acid called methionine. Okay? Okay, that leaves us 60 left, and guess what? We only have 20 amino acids that go into proteins. Okay, so what happens here, we've got 20 amino acids, we have 60 more possible codons. So what happens here is many amino acids are specified by more than one codon. There are a few amino acids, and these tend to be the least abundant in proteins, where we have one single codon that specifies that amino acid. Most of them you have like 2 or 3 or 4. Glycine, the simplest and most abundant amino acids in proteins, have 7 separate codons that code for that. Another thing we see with the genetic code here is many times, if you have something like a lot of times in this case, I'm not sure if this is specifically the case for this example, this is representing. A lot of times if you have these codons, if you change the last base, many times that codon still represents the same amino acid. So a lot of times you have things where a single point mutation is not going to change the amino acid at all and introduce a mutation you don't want into the protein. Or if it does alter the amino acid, many times you're going to stay in the same class of amino acids. Like a polar amino acid gets substituted with another polar amino acid. That is not always the case. A good example of that is sickle cell anemia. Because in this case, yeah, it is. Just want to make sure. Yeah, okay. Sickle cell anemia, in this case, there is a single point mutation changing one codon in the beta globin gene. And unfortunately, that change takes a polar amino acid and substitutes it with a hydrophobic amino acid. That causes a change in the globin structure, and it causes the hemoglobin molecules to stick together to form these rod-like structures that puncture and bend and distort red blood cells, causing the symptoms of disease. The red blood cells are destroyed a lot faster than they would in normal people, and on top of that, distorted red blood cells clog up capillaries and things like that and cause all kinds of problems. So that's where you get the clinical symptoms of sickle cell anemia. And of course, that's found predominantly in people of Central African ancestry. There is something called the beta thalassemias that's very similar, and that's found in people of Mediterranean ancestry, like Greek, Italian, stuff like that. It turns out that people that are heterozygotes for either the thalassemia or the sickle cell trait are phenotypically normal in the sense they don't get sick, but they're many-fold resistant to malaria infection than people who are completely the normal or wild type thing. So that's why these anemias, that's why these diseases stay at fairly high levels in the populations and stuff. If you look at, say, the incidence of the sickle cell trait in the genes and stuff, and the malaria risk, they coincide very closely. Central tropical Africa, where you have very high rates of both. Other parts like northern and far southern Africa, you have it in much lower proportions. So that's sort of an adaptive thing. So we see in effect in the genetic code, we see some kind of evolutionary adaptations where you can make an occasional point mutation and not necessarily change the protein, at least change it all that much. Now nobody knows, the genetic code we see here is pretty much universal. There are a few exceptions, a couple mitochondria in a few organisms, one codon actually represents something different than the standard genetic code. But those are extremely rare exceptions. They appear to have happened somewhere later on in evolution. But for the most part, the genetic code, what codons represent what amino acids, is the same in the lowest archaean as it is in us. It's something universal. Now, as the origins of this, it's possible early on, before there were signs of life in the fossil record, there may have been multiple genetic codes, and for some reason or another, one of them happened to win, and we're all descended from that. That's sort of some hypothesis and stuff when you go back close to 4 billion years ago, the origin of life on this planet. Okay, but we pretty much use the same genetic code now. All right, now, it may bring up a question here. Why does the genetic code, why does a sequence of a codon, a sequence of 3 bases, why does that codon represent a particular amino acid? For instance, why does the codon UUU represent the amino acid phenylalanine? Why doesn't it represent something else? Glycine or lysine or serine or whatever. Well, how does that work? Well, that is where we have our next major character in protein synthesis, the transfer or tRNAs. Now, tRNAs are critically important for the process of translation, because what they do is they essentially carry a specific amino acid to the ribosome, put it in a specific place, and then the ribosome can join these things together. So, now tRNAs are small RNAs. They form, if you look at it, they have regions that are hydrogen bonded to each other, what we call stem regions, and regions that are not, which we call loop regions. So, in kind of 2 dimensions, a tRNA looks like a 4-leaf clover. You have a section here, and hydrogen bonds over here. We have an amino acid, I'll call it AA, that's covalently attached to this end here. And now we have like a traffic circle, and then we have another region, a stem region that's hydrogen bonded, and a loop region that's not. And we have another region over here, the stem and loop region. And then we have another one over here. So it kind of looks like a 4-leaf clover. Okay, that's the basic structure of tRNA. They're about 130 or so nucleotides long. They're relatively small RNAs. And there are all kinds of different tRNAs. All tRNAs carry a specified amino acid at this particular end. Now that amino acid is covalently attached to it by a type of protein called the aminoacyl tRNA synthetases. It's a jawbreaker, right? What these are protein enzymes that recognize a specific tRNA, bind a specific amino acid, never the wrong amino acid and never the wrong tRNA, and then they covalently attach the amino acid over to here. So there are as many of these amino-acyl tRNA synthetases as there are tRNAs. And because attaching amino acid like that, that's an endergonic condensation process, these guys have to use a source of energy. I believe it's GTP hydrolysis to do that. Okay, so these are what actually attach the amino acids to the tRNAs. Now we look at this, and there's one especially critical part of the tRNA, which right over here, at a particular part of this top loop, there's a sequence of 3 bases called the Anticodon. This is a continuous nucleotide sequence, but 3 of these nucleotides in a particular thing are called the Anticodon. Okay, and that is the part that will bind and form hydrogen bonds with the appropriate codon of a messenger RNA. So let's take a look at this. We're going to have part of a messenger RNA. Right here we have the codon UUU. The UUU stands for phenylalanine. Well, you happen to have a tRNA out there that has an AAA anticodon. That means we can make hydrogen bonds between them, 2 hydrogen bonds each, and that holds the thing in. And that UUU tRNA always carries the amino acid phenylalanine. It never carries another amino acid. Now, there would be, for glycine, for instance, there would be 7 tRNAs with 7 different anticodons, and they would recognize and hydrogen bond to the 7 codons that represent glycine. In other words, the reason that UUU represents phenylalanine, that's the only one I know offhand. Okay, the UUU represents phenylalanine is that codon UUU, there is a tRNA with the anticodon AAA, and that tRNA always carries phenylalanine, never any other amino acid. Okay, well, the reason that the codon UUU, we said it represents phenylalanine, the question we have here is why phenylalanine, not something else? Okay, the reason is the tRNA. The reason is that there is a tRNA out there that's its perfect match. That tRNA happens to have the anticodon AAA, right at the exact location it needs to be. So it can hydrogen bond to the UUU codon. And that tRNA, the one with AAA, the only amino acid it will ever carry is phenylalanine. It never carries another. So the genetic code, what codons represent what amino acids, is because that there are tRNAs with the appropriate anticodon, and they carry that and only that amino acid. So tRNAs are absolutely critical for the process of translation. Now, by the way, in 3 dimensions, this is kind of like, this has been splayed out, you know, the crucified tRNA, right? Okay, it's been splayed out like it's getting crucified. Actually, come down to it. When a tRNA binds the ribosome, that crucifixion analogy is not so bad. When the tRNA binds the ribosome, you're talking about codon and anticodon binding. That's 6 hydrogen bonds. That ain't much. But those side arms bind to part of the ribosome and it holds it in place. It stabilizes it. So saying about the crucified tRNA is not all that inaccurate. It's those side arms that nail it to the ribosomal cross in effect. It really stabilized that because it was just those 6 hydrogen bonds, I think, would fall off in a moment's notice. So the side arms are involved in binding to the appropriate part of the ribosome. Okay, so that's a major player here in the process of translation, those tRNAs. Now, by the way, organelles, mitochondria, and chloroplast, they make their own tRNAs. As a matter of fact, the tRNAs are the prokaryotic type because they are descended from cyanobacteria or proteobacteria, depending on what organelle you are. So they have the same kind of machinery. They do make their own tRNAs. They don't have to rely on imported tRNAs in the cytoplasm. They like their own prokaryotic tRNAs. Okay, well, and they got the genes for them too. Okay,